Hello, everyone. I am Trishla from University of Hyderabad, India. And today I'll be talking about the modular nature of transcription factors with a particular example of a cotton transcription factor from a cotton transcription factor GH-NAC4 with regards to ABA and abiotic stress responses. Before I begin, I would like to give a brief introduction about NAC transcription factors. These are one of the largest plant-specific transcription factors with more than 100 members, and they consist of two domains. Typically, they consist of two domains, a conserved N-terminal NAC domain and a C-terminal transcription regulatory domain. And they are known to play an important role in various plant growth and developmental processes such as shoot apical meristem, seed development, flower development, leaf senescence, and also in environmental stress responses such as abiotic and biotic stress. Before I begin, I'd like to talk about a very interesting experiment that was carried out by Brent and Tashney in 1985. It's called the domain swap experiment, where they took the DNA binding domain of a repressor protein from E. coli called LEX-A, and they fused it to the activation domain of a transcription factor from yeast called as GAL4. And that resulted, that chimeric protein identified LEX-A binding site, but acted as a transcription activator. Now, this was also replicated in various other systems, and it was concluded that transcription factors have this domains which can act as a different independent structural modules with independent functions. Now, this similar modular nature of transcription factors was also seen for NAC transcription factors. A study published by Jensen et al. In 2010, they swapped the N-terminal domain with the N-terminal of a transcription factor called as ANAC19 with the N-terminal domain of various other transcription factors. And the same thing they did with the C-terminal domain. And they studied the response to a stress hormone. Plants produce this hormone called as abscisic acid. So in response to that hormone, they studied, and they've concluded that the N-terminal domain was responsible for the response to this phytohormone abscisic acid for this particular NAC transcription factor called as ANAC19. A similar study by Taoka et al. Was published in 2004, where they studied the domain swap analysis of a CUC transcription factor, CUC. These transcription factors are known to play essential role in shoot apical meristem development and shoot induction. So when they did domain swap analysis, they identified that the N-terminal domain was essential for the shoot induction functionality, while the C-terminal domain could be swapped with other C domains of other NAC transcription factors. But so far, NAC transcription factors' response to abiotic stress has not been studied. This structure function relationship of NAC transcription factor in response to abiotic stress tolerance has not been studied. So we wanted to analyze that. And we chose a particular NAC transcription factor from cotton because the study by Meng and co-workers in 2009 have identified that a particular NAC transcription factor called as GH-NAC4 was highly upregulated by drought, stress, salt stress, cold stress, and also by ABA. So we thought that this is an ideal candidate to study this abiotic stress response. And so we first went ahead and did expression analysis in response to various hormones. And we observed that this particular NAC transcription factor was highly upregulated by abscisic acid, by salicylic acid, by gibberellic acid, by methyl jasmonate and ethylene, also by cytokinins and oxalins. And the expression levels were also highly upregulated by drought stress, by salt stress, by osmotic stress, oxidative stress, by heat, by a combination of cold and dark treatment, and also by wounding and air drying. So after this, we analyzed the promoter sequence of this particular transcription factor. And we isolated 1500 base pair upstream. We predicted the transcription start site, and we identified all the cis-acting elements present in this promoter, and we observed that there's several motifs required for uh, hormone responsiveness, for stress responsiveness, for tissue specificity, for light responsiveness were present in the promoter region. And here is the physical map showing the location of the cis-acting elements of the promoter region. After analyzing this, we were curious to see the spatio-temporal localization of this promoter. And for that, we fused the promoter with a reported gene called as GUS, and we transformed this in tobacco plants, generated transgenics, and then we analyzed the spatio-temporal localization. We observed that the GH-NAC4 promoter was localized to the radicle, the tip of the radicle, in the growing cotyledons, in the shoot apical meristem part, and also in the root part. And interestingly, in 7-day-old seedlings, we observed that the localization was present in the cotyledons, the shoot, and the root, but it was absent in the hypocotyle region. In a 15-day-old seedlings, we see that the GH-NAC4 was localized almost throughout the plant, especially in the shoot apical meristem, the stems, the roots, and the midrib of a young leaf, the lateral veins, and also the guard cells. Coming to the root part, we see that it was present in the root. It was present in the main root, and it was also present in the lateral root, but it was absent in the root cap region and also in the root hairs. Staining of the floral structures have identified the localization in the sepals, in the edges of the petal, in the stigma, the anther, and also in the pollen grains. Now, to understand the tissue specificity of GH-NAC4, further what we did was we made freehand cross sections using a laser braid of the leaf vein, the petiole, the stems, and the roots. And we observed that the localization was specifically present in the vascular bundles. And in vascular bundles, it was specifically present as in the phloem region. Both the abaxial and the adaxial phloem region seem to be the site of localization of GH-NAC4. As you can see in here, roots and in stem and petiole and the leaf veins. And the cross-section of a wounded stem showed that intense staining was also observed in the other tissues like the epidermis and the cortex. After understanding the tissue localization, we wanted to see the subcellular localization of this particular GH-NAC4 transcription factor. And for this, we used an interesting technique called as Agro infiltration. In this technique, we take agrobacterium, which is transformed with a particular vector of our interest, a plasmid of our interest, and that is injected into the interstellar spaces of a tissue, like a leaf, and the agrobacterium goes on and infects the neighboring cells. So agroinfiltration of a plasmid where this GH-NAC4 is fused to a GFP shows that it is predominantly localized in the nucleus. Then we went ahead to study the structure of GH-NAC4. And we observed that GH-NAC4 is a typical structure of a NAC transcription factor with the N-terminal NAC domain and a C-terminal domain. Now, the C-terminal domain was predicted to possess intrinsically disordered regions. Now, these intrinsically disordered regions are low complexity regions which have a high composition of polar and charged amino acids. And so we wanted to see if these domains play any separate role in the function of GH-NAC4. So for that, to study if this particular protein GH-NAC4 acts as a transcription factor, we performed an assay called as the transactivation assay in yeast. Now, what is this transactivation assay? In transactivation assay, a protein of our interest is fused to the DNA binding domain of a particular protein called as GAL4. And if this protein of our interest is able to recruit the RNA polymerase to the transcription site, there'll be expression of the reporter gene, which will lead to the growth of the cells on a selection media. Now, if that particular protein has a transactivation property, only then we will see the growth of the yeast cells. So we see here that the full length protein was able to show very high levels of transactivation, while the end domain could not grow at all. The C terminal domain also had very high level of this transactivation activity, and here is we did a deletion analysis of the intrinsically disordered regions that we had predicted earlier, and we see that these regions are also essential for the transactivation property of GH-NAC4, and that the C terminal domain is what is the C terminal domain has the property of transactivation or the transcription regulatory property. A study by Ernst and co-workers in 2004 showed that these transcription factors also exist as homodimers or heterodimers. As they showed that these particular two salt bridges formed by conserved amino acids are responsible for the dimerization of GH-NAC4 transcription factors. So we were interested to study if if GH-NAC4 was also forms homodimers. So for this, we used an assay called as yeast-2 hybrid assay, where a protein of interest, let's say X, is bound to the DNA binding domain, and another protein, let's say Y, is bound to the activation domain. Now, if these two proteins interact with each other, then the DNA binding domain and the activation domain are brought closer to each other, which leads to the expression of the reporter gene, which is analyzed as growth of yeast cells on selection media. So when we perform this, we see that the N-terminal domain was able to homodimerize, while the C-terminal domain was not able to, and any point mutation to disrupt the salt bridges led to the abolition of the homodimerization. Now that we've studied the biochemical properties of the different domains of GH-NAC4, we were interested to study the physiological function. Now, for that, we generated tobacco transgenics of the full-length transcription factor, the N-terminal domain, and the C-terminal domain. And what we started by what is called as the seed germination assay. Now, in this assay, seeds of the wild-type plants and the transgenic plants were sown on selection media, on a stress media in this case, and were allowed to germinate for 7 days. And after that, the germination percentage was scored. We see that under salt stress, both the transgenics and both the domains were able to germinate as compared to the wild type. And under drought stress also, the transgenics and the wild type, the transgenic, the full-length transgenics and the N and C terminal domain transgenics showed better germination percentage as compared to the wild type. Now, on a media supplemented with ABA, which is the stress hormone that I mentioned earlier, abscisic acid, the N-terminal domain transgenic showed very poor growth as compared to the other plants, and while the C-terminal domain showed very high percentage of germination. Now, with this assay, we see that the GH-NAC4 has stress tolerance capabilities, and both the domains add to that stress tolerance capability. And specifically, this N-terminal domain shows sensitivity to AB. To corroborate this data, we went ahead and performed an assay called as the roots length assay. where 7 days old seedlings were transferred to were transferred to stress media and allowed to grow for 15 days. And we see that under both salt and drought stress, the transgenic plants had a higher root length as compared to the wild type plants. And under ABA, in ABA media, the N-terminal domain transgenics had very poor growth. This further confirmed our hypothesis that the both the domains are responsible for the stress tolerance capabilities of GH-NAC4, while the N-terminal domain was responsible for the ABA receptivity. Later, we went ahead to study what is called as this stomatal behavior. So before that, I'd like to briefly explain about it. It is well known that stomatal movement is largely controlled by the phytohormone that I explained earlier called abscisic acid. And measurement of stomatal aperture is a useful indicator of altered ABA sensitivity. As we've seen earlier, that GH-NAC4 transcripts were upregulated by ABA. And when we characterized the promoter, we had seen that the guard cell specific motors are present in the promoter region, and also the promoter was localized to guard cells. Now, this prompted us to study the stomatal behavior under ABA stress, under ABA treatment. Before we do that, we observed that under controlled conditions, the transgenics of both the N-terminal domain and the C-terminal domain had smaller aperture as compared to the wild type plants. And under ABA stress, all the plants showed stomatal closure, which was expected, but the N-terminal domain had a very small aperture, had like higher percentage of closure as compared to the full length protein and the C-terminal domain and the wild type plant. Now, with this, we conclude that the ABA hypersensitivity resides with the N-terminal transgenics, and the C-terminal transgenics are nearly insensitive. Here is the graph showing the quantification of the stomatal aperture width under ABA treatment. After studying this differential behavior of the transgenics to ABA, we wanted to see if they also showed difference to stress tolerance. So for this, we performed an assay called as the water loss assay. In this, leaves were excised from the wild type and the transgenic plants, and the water loss assay was examined over a period of time. And after 9 hours, we see that the wild type plants have lost a lot of amount of water as compared to the transgenics, and the C-terminal domain transgenics did not fare as good as the N-terminal domain or the full-length transgenics. Now, this could partially be due to the inability to close the stomata, as we've seen earlier, the inability to close stomata because of insensitivity to ABA. Here is the graph showing quantification of the water loss by the transgenics and the wild type over a period of time. Next, we performed what is called as the leaf disc assay to analyze the stress tolerance capabilities. In this assay, what we did was we leaf discs of the transgenics and the wild type plants were floated on different solutions for 4 days, and then the biochemical parameters were analyzed. And as we see here, that the wild type plants showed a high amount of bleaching as compared to the transgenic plants. And this shows that GH-NAC4 is involved in adaptation to salinity and drought stress, and both the domains are involved in stress tolerance. Next, we performed what is called as the lipid peroxidation assay. Now, under abiotic stress like drought and salinity, lot of reactive oxygen species are generated, which leads to the peroxidation of the lipids, especially the plasma membrane and other endomembranes. Now, that is analyzed by an assay called as the T-bars assay, where the malondialdehyde content of the cells is measured, of the tissue is measured. And we see that the wild type plants had a very high content of MDA, as compared to the transgenic plants. Next, we analyzed we analyzed the proline accumulation. Now, proline are the osmolytes which plants produce to maintain a redox homeostasis under stress conditions. We observed that the transgenics had a higher extent of proline accumulation as compared to the wild type plants, and the full-length protein had the highest amount of proline accumulation among all of them. Now, given the differential regulation of stress tolerance, it is important to question whether there is any alteration of the stress responsive genes in the transgenics upon drought and salt stress. So we investigated the expression levels of various antioxidant enzyme transcripts under salt and drought stress. We studied the expression of ascorbate peroxidase, catalase, and superoxide dismutase. And we observed that under both the salt and the drought stress, there was a very high upregulation of transcripts of these antioxidant enzymes in transgenic plants as compared to the wild type plants. Next, we also studied the expression of various other well-known stress responsive genes, such as ERD10. Now, this ERD10, these are osmotic hydrophilic proteins which play a role in redox homeostasis. And another gene that we studied was ethylene response factor 5. This is known to play a crosstalk in hormonal and stress regulation. And the third gene that we studied was DREP, which is DRE binding protein. Now, these play a very important role in regulation of stress responsive genes and in ABA independent pathway. In both the three cases, we saw that there was a very high upregulation of the transcripts in the transgenic plants as compared to the wild type plant, especially in the case of this DREP transcription, transcripts, where more than 100-fold upregulation was seen in the full-length transgenics as compared to the wild type and the other domain transgenic. Now, the expression of NCED, SOS1, and SUS1 was also studied. NCD is an ABA biosynthetic gene. SOS1 is a plasma membrane sodium proton exchanger, and SUSI is a sucrose synthetase gene. And as we've seen earlier, high upregulation of these stress responsive genes was seen in transgenics as compared to the wild type, and a very high upregulation of this ABA biosynthetic gene was observed. So with this, I'd like to conclude that the stress tolerance capabilities of GH-NAC4 is associated with both the component domain, while the ABA responsiveness is largely associated with the N-terminal NAC domain. And the N-terminal NAC domain is responsible for the homodimerization of GH-NAC4, while the C-terminal transcription regulatory domain is responsible for the transactivation of GH-NAC4. Now, this part of the work was published in these two papers, and with this, I'd like to conclude and like to acknowledge that this work was done in the laboratory of Professor P.B. Kirti at the Department of Plant Sciences, University of Hyderabad. And I'd like to thank my collaborators, Dr. Prasilla, Dr. Suresh, Mr. Bharat, Dr. Ashwini, and Mr. Venkat. I'd like to also thank Professor Krishnaveni for giving the yeast vectors for the yeast assays. And a big thanks to the PCA Online Summit organizers for giving me this opportunity to speak. And if anybody would like to contact me for further details on the experiments that I've performed, you're most welcome to email me or connect with me on LinkedIn.
Thank you, Dr. Trishla, for your wonderful presentation, and that is so much of research. And I will leave some time here. If anyone of you have any questions or comments, you can please go ahead and ask Dr. Trishla.
Hello, Dr. Trishla.
Hello, Prasanna.
Hi, Trishla. And I just want to congratulate you to do this great work. Just section the transcription factor and just studied in and around of this function of the transcription factor, the structural level and functional level by producing transgenics is a very great work. I just like to congratulate you for doing this job and all the best for your career.
Thank you very much. Thank you very much.
Dr. Yan, you wanted to ask some question?
Yes, Dr. Yan.
It's me. Hi.
Hi.
And very nice work.
Thank you very much.
And I have one question about the transgenic plants only expressing the N terminal of the neck.
No,
so the transgenic plant only express with the N terminal of the neck transcription factor. It shows that it's highly sensitive to ABA, right?
Yes.
But somehow the expression pattern of this mutant seems not so different compared with other transgenic plants in your expression pattern analysis.
You mean the expression analysis was for various stress responsive genes in the transgenic plants?
Yes.
Not as compared to the transgenic plants, but in the transgenic plants, because this NAC is a transcription factor, so we believe that it would alter the expression of various other stress responsive genes. NAC transcription factors usually are at a very early stage response genes. So that's why we study the expression of various other genes in the transcription factor, in the transgenics.
Yeah, but how do we explain the hypersensitive response to the ABA in the N-terminal transgenic plants?
Yes. So the full-length protein itself is slightly sensitive to ABA. We call it what is called as the positive regulator of ABA. It's getting upregulated by ABA. It has ABA responsive motifs. So that ABA receptivity of the full-length protein resides in the N-terminal domain. And since we overexpressed, we expect that because it is sensitive to ABA, because of overexpression, that led to the hypersensitivity.
Okay. So in the in the last part of your talk, you check the expression pattern of these responsive genes. So have you done the treatment under ABA treatment? How does gene expression?
No, we checked under salt and drought stress. We did not check the expression levels of these genes under ABA stress. We did not under ABA treatment. We did not do that. We checked under salt and drought stress.
Okay, thank you. No more question.